For centuries, the ultimate human endeavor was mapping the globe. I mean, explorers would set out into the absolute unknown, sketching coastlines, navigating deadly storms, dropping anchor in strange bays, and just slowly filling in the blank spaces on the map.
Yeah, it was this massive generation-defining project that completely changed how we understood our place in the world.
Exactly. But today, we are looking at a completely different kind of blank space. We are turning our attention inward to the ultimate biological cartography. We've got a stack of research notes and slide decks here on genome mapping, and we're taking a deep dive into the blueprint of life itself.
It really is fascinating.
Right, and whether you are prepping for a biology exam or you're just insanely curious about what actually makes you, we are about to decode how scientists navigate this microscopic landscape.
It's the modern equivalent of mapping a new continent, really.
Mm-hmm.
But instead of mountains and rivers, we're navigating 3 billion base pairs of DNA.
Which is just a wild scale to think about.
It is. And our mission for this deep dive is to understand exactly how scientists chart this territory based on the source material.
Yeah.
Like, how do they locate the specific genes associated with diseases? How do they trace our evolutionary history?
Right.
We're going to explore the incredible technology of how we actually build this map. And eventually, we're going to look at the surprising, shape-shifting nature of DNA itself.
Okay, let's unpack this. Because when you first think about mapping a genome, the immediate thought is, well, I always wonder, is this basically just Google Maps for our DNA? You open an app, type in breast cancer gene, and it drops a little red pin on chromosome 17.
I mean, that was a great analogy, actually. But to make that analogy accurate, you have to realize that this map has two completely different views.
Okay.
Think about how your map app has the standard street view and then it has the satellite view. In genome mapping, we have genetic maps and physical maps.
Wait, so what is the street view in this case?
That would be the genetic map, which is sometimes called a linkage map. Genetic maps don't actually measure physical distance. Instead, they measure how often traits are inherited together.
Oh, interesting.
Yeah, they use a unit of measurement called centimorgans, which measures recombination frequencies. So it basically tells you, hey, the gene for eye color and the gene for this other specific trait tend to travel together during reproduction. But it doesn't tell you exactly how many molecular miles apart they are.
Right, so it's relative. It's like giving someone directions by saying, the coffee shop is about a 5-minute walk from the bookstore.
Exactly.
It's helpful, but it's not a precise GPS coordinate.
Right, exactly. Now, physical maps, on the other hand, show the absolute concrete physical distances. This is your satellite view.
Okay.
They measure the distance in base pairs, so the actual chemical rungs on the DNA ladder. You absolutely need physical maps if you want to sequence a genome, and they are essential when you're trying to zero in on the precise molecular location of a disease-associated gene to develop a treatment.
So to build this ultimate physical GPS map, scientists have to sequence the DNA. They have to actually read the letters, A, C, T, and G. But the immediate problem there is the sheer scale, right?
Oh, yeah.
The human genome is massively long. You can't just put an entire chromosome under a microscope and read it continuously from start to finish. You have to break it apart first.
Yes, you have to dismantle the puzzle before you can figure out how the pieces fit together.
Yeah.
And historically, there have been two main ways to tackle this assembly and sequencing phase.
Okay.
The first is called the clone-contig approach. Think of this as the highly meticulous step-by-step method. The genome is first mapped out into large, manageable chunks called contigs.
So you're grouping things into neighborhoods first.
Precisely. You isolate a specific contig because you already know roughly where it belongs on the larger map. Then you break that specific chunk down further into tiny fragments and put them through what's called shotgun sequencing. You sequence those tiny fragments, and because you already know their original neighborhood, putting the tiny pieces back together is relatively straightforward.
Because they all belong to the same block. You aren't like mixing pieces from New York with pieces from Tokyo.
Exactly. Now contrast that with the second method, which is the whole genome shotgun approach. This is the fast, computationally heavy method.
Right.
Instead of carefully mapping out neighborhoods first, you just shatter the entire genome randomly into millions of tiny fragments. You sequence all those fragments directly, all at once.
Just massive chaos.
Absolute chaos.
Yeah.
And then you rely on massive supercomputers to find the overlapping regions at the ends of these fragments to piece the entire thing back together.
It's wild. So the whole genome shotgun approach is like throwing a 1,000-piece puzzle of a perfectly clear blue sky into a blender and trying to piece it back together.
Yes, pretty much.
Without the picture on the box, those identical blue edge pieces are going to cause some serious incorrect overlaps.
That is the exact problem they run into. Both methods, but especially the whole genome shotgun approach, they struggle massively with repetitive DNA. Because, I mean, our genome is just full of repeating sequences.
Right, the identical blue puzzle pieces.
Exactly. So when the supercomputer is looking for overlaps to stitch everything together, those repeating sequences completely trick the assembly process. It might see, a sequence like GATTA on two different fragments and just assume they overlap.
When they really don't.
Right. In reality, they might be from completely different chromosomes that just happen to have the same repeating pattern. It creates incorrect overlaps and false assemblies.
So if identical puzzle pieces are ruining the assembly, how do scientists actually put this thing together without making a mess? Like, how do they avoid getting lost in those repeating patterns?
They need incredibly stable, unique landmarks. They essentially drop anchor pins on our digital map to orient themselves. And one of the most important landmarks they use is called an STS, or a sequence tag site.
What makes it a landmark exactly?
So an STS is a short piece of DNA, usually just 200 to 500 base pairs long. But what makes it special is that this specific sequence of letters appears only once in the entire genome.
Wait, really? Only once out of billions of letters? That is the ultimate anchor.
It really is. Scientists use a process called PCR to copy and screen for these STSs across a massive library of overlapping DNA fragments. If two completely different fragments both contain the exact same STS, it proves without a doubt that those two fragments overlap in the genome.
Makes sense.
By finding enough of these shared STSs, scientists create a linear scaffold. They literally map out the overlaps to build the genome structure.
Okay, so that maps the identical parts, the scaffolding of a human being. But humans aren't clones. We all look different. We respond to diseases differently. How do scientists map the mutations? How do they map the differences between you and me?
To find those differences, scientists use something called restriction enzymes. You can think of them as highly specific molecular scissors.
Okay, I like that visual.
These enzymes scan the DNA strand, and they only make a cut when they find a very specific sequence of letters, which is what we call a restriction site. Now, some of these cutting sites are exactly the same in every single human being. We call those non-polymorphic sites.
Right, because poly means many and morph means form. So non-polymorphic means it doesn't change its form. It's stable across everyone.
Right, exactly. But other cutting sites vary from person to person. Maybe you have a mutation or a deletion and the sequence has changed. These are polymorphic restriction sites. This gives rise to a technique called RFLP, restriction fragment length polymorphism.
Okay, so let's say we're comparing my DNA to your DNA, and we're looking at a specific gene. My gene might have that specific cutting site intact, but your gene might have a tiny mutation right at that exact spot.
Exactly. So when we apply our molecular scissors to your DNA, it doesn't recognize the mutated site, so it skips it. It might only cut your DNA strand into 3 fragments.
Oh, I see.
But when we apply those same scissors to your exact same gene, because your cutting site is intact, it cuts it into 4 fragments. By simply visualizing the number and the size of the fragments, we can instantly detect the genetic difference between us.
But wait, if I hand you 3 pieces of string that I just cut, you know their lengths, but you don't know what order they're in. You don't know which piece of string was originally in the middle. If a single enzyme only tells you the fragment sizes, how do you map their actual sequence?
That is a brilliant question, and it's where the logic puzzle of genome mapping comes in. To find the order, scientists use a double digestion. Instead of using one pair of molecular scissors, they use 2 different restriction enzymes at the same time. Let's call them enzyme A and enzyme B.
Okay, walk me through this, because the logic here is fascinating. Let's say you start with a long piece of DNA.
Okay, well, imagine a DNA fragment that is 4,900 base pairs long. When you cut it with enzyme A alone, one of the pieces you get is 700 base pairs long.
Sure.
But when you do a double digest, meaning you apply enzyme A and enzyme B at the same time, that 700 base pair piece suddenly disappears.
Just vanishes.
Yep. And in its place, you find a 200 base pair piece and a 500 base pair piece.
Ah, which proves there must be a cutting site for enzyme B hiding inside that specific piece created by enzyme A.
Exactly. It's pure deduction. By comparing the fragment lengths from the single digestions to the double digestions, scientists can logically deduce the relative position of every single cut site. It is literally biological Sudoku.
That is incredibly clever. And as technology evolved, the tools for finding these variations got even faster, right? For example, the sources mention SSLPs, simple sequence length polymorphisms.
And that's a big one.
Here, scientists look at regions of DNA that just repeat the same letters over and over. They copy these regions and then run them through a process called capillary electrophoresis. They basically put the DNA fragments into a microscopic tube, apply an electric current, and make the fragments race to the finish line.
And because DNA has a negative charge, the electric current pulls it along. The shorter fragments zip through the tube much faster than the longer ones.
Naturally.
So just by timing the race, scientists get a hyper-precise measurement of how long your specific repeats are.
But the holy grail of finding these variations is detecting SNPs, single nucleotide polymorphisms. These are variations of just a single letter in the DNA code. Finding a one-letter difference in a sequence of 3 billion letters is a monumental task.
It is the ultimate needle in a haystack. To do it quickly, scientists use a brilliant technique involving a tracker molecule that utilizes dye quenching.
Here's where it gets really interesting, because they essentially create a molecular light switch. The tracker molecule has a fluorescent dye attached to one end and a quencher molecule attached to the other end.
Right.
When the tracker is just floating around, it's curled up. The quencher physically hugs the dye and absorbs all its energy, keeping it completely dark.
Yeah, it acts like a shade over a lamp.
So how does the light actually turn on? What happens when it finds the specific genetic mutation it's looking for?
It's all about physical shape. When that curled-up tracker molecule encounters the exact, perfectly matched single-letter mutation it was designed to find, it binds to the DNA strand. But the physical act of binding forces the tracker to snap perfectly straight.
And when it snaps straight, the quencher is physically pulled far away from the dye.
Exactly. The shade is removed. Once they're separated, the fluorescent dye can suddenly emit light. A perfect genetic match literally flips the switch and the molecule glows.
That's amazing.
If the genetic sequence isn't a perfect match, the tracker stays curled up, the quencher stays close, and the sample remains completely dark. It allows doctors to detect specific genetic variants in real time just by looking for a glow.
That is so elegant. But taking a step back, there's a huge hurdle we need to cross. We've been talking about single letters and tiny 500 base pair fragments, but the human genome is huge. Entire chromosomes are millions of base pairs long, megabases. How do you map the really big stuff? Because standard laboratory tools just break down at that massive scale.
They absolutely do. Take standard agarose gel, which is the traditional way scientists separate DNA by size. You place the DNA in a jelly-like matrix, turn on a constant electric field, and pull the DNA through. Small pieces sieve through the pores of the jelly easily, but pieces of DNA larger than 50 kilobases just hit a wall.
Why do they hit a wall?
Because the pores in the gel are too small.
Yeah.
At that massive size, the DNA molecules basically squish into a shape that just continuously slides through the gel at the exact same agonizingly slow speed. You completely lose the ability to separate them by size.
So what does this all mean for mapping? If standard gel is like cars driving on a highway, a compact car and a massive semi-truck can both drive straight forward at 60 miles an hour. If you just watch them drive straight, you can't separate them by their mobility. So scientists invented OFIG, orthogonal field alternation gel electrophoresis.
It's a mouthful, but the concept is brilliant. Instead of a constant single-direction electric field pulling the DNA straight ahead, OFIG uses an alternating electric field. It constantly changes the direction of the pull at regular intervals.
Which changes the highway into a tight alleyway.
Right.
The electric field flips. A compact car zips around and reorients to the new direction quickly. But a massive semi-truck takes forever to execute that multi-point turn before it can move forward again. By constantly changing the direction, OFIG separates massive DNA molecules based entirely on how long it takes them to reorient themselves.
That is a perfect visualization. The larger the DNA molecule, the longer it takes to reposition itself to the new electric field, which slows its overall progress through the gel. This allowed scientists to finally separate and map entire chromosomes, extending into the megabase range.
Incredible.
But sometimes you don't want to run things through a gel. Sometimes you just need to directly look at these massive DNA giants.
How do you look at DNA? It's crumpled up inside the cell like a ball of yarn.
You have to unspool it. One technique is called molecular combing. It sounds like something you do at a salon, but it's highly precise. They take a specially coated piece of glass, think of it as molecular Velcro, and drag it slowly through a solution of unspooled DNA.
Okay.
One end of the DNA strand attaches to the glass, and as the glass is withdrawn, the surface tension of the receding liquid stretches the DNA strand perfectly straight, laying it flat, just like combing wet hair.
And once it's stretched flat, they can tag it with fluorescent markers and actually look at the physical restriction sites directly under a microscope. It's called optical mapping. And they are making this even faster using microfluidics. They build high-throughput microchips with microscopic channels and use highly controlled fluid flows to stretch thousands of DNA molecules out simultaneously.
But stretching the DNA pulls it out of its natural context. If you want to see where a gene lives on the actual intact chromosome, you use fluorescence, in situ hybridization.
In situ, meaning in its original place.
Exactly. They take chromosomes while they are perfectly packed together, gently unzip the DNA strands right on the microscope slide, and apply fluorescent probes. Those probes hunt down their specific matching sequence and bind to it.
Wow.
So when you look under the microscope, the exact physical location of a gene literally lights up as a glowing dot on the structure of the chromosome itself.
It's a literal map with a "you are here" beacon. But the technology doesn't stop at finding genes. Scientists are now building their own maps with light-activated synthesis. This isn't just reading DNA. This is building custom DNA sequences right on a tiny glass chip to create microarrays.
It works a lot like printing a highly complex photograph layer by layer. The chip is covered in light-sensitive protective chemicals. Scientists shine a highly targeted microscopic light on very specific spots on the chip, which removes the chemical shield in those spots only.
Right.
Then they wash a single DNA letter, say adenine, over the whole chip. But it only permanently attaches to the unshielded spots.
Then they reapply the shield, shine light on different spots, and wash it with cytosine. By repeating this process layer by layer with A, T, C, and G, they can synthesize thousands of custom DNA sequences in parallel. This is how we manufacture the gene testing chips used in hospitals today.
It is the pinnacle of modern diagnostics. But there is a massive paradigm shift hiding in the final section of this research.
Oh, yeah.
Everything we have talked about so far, the mapping, the cutting, the stretching, it all relies on one core assumption. It treats DNA like a rigid, fixed ladder. We assume it only ever fits together one specific way.
The classic double helix, Watson and Crick.
Right. We are all taught about Watson-Crick base pairing. Adenine always pairs with thymine. Guanine always pairs with cytosine.
Mm-hmm.
It is the gold standard for storing genetic information. It is incredibly stable and predictable.
But the research introduces something called Hoogsteen base pairing. It turns out the building blocks of DNA aren't entirely locked in place. The larger bases, the A and the G, can actually rotate. They physically twist around and use completely different atoms to form bonds.
Yep.
And when they do this, they don't just form a double helix, they can create a triple helix. I have to say, I was always taught Watson-Crick was the only way DNA fits together. Are you saying our biology textbooks left out a secret backdoor into our DNA structure?
Yes, a backdoor that fundamentally changes how we view biology. Hoogsteen pairing isn't a mistake. It's a critical dynamic feature of our genome. Watson and Crick map the most stable state of DNA, but they missed this because Hoogsteen pairing is temporary and shape-shifting.
Why would our DNA need to temporarily change its shape?
For survival. It plays a massive role in gene regulation. Some proteins in our body bind to the DNA and actively force it to switch from Watson-Crick to Hoogsteen pairing. This structural switch literally bends the DNA, making it impossible to read. It effectively turns a gene off.
Oh, wow.
It's also vital for DNA repair. If DNA is damaged by toxins, Hoogsteen pairs can temporarily form to help the cell's repair machinery recognize the error and fix it.
So the blueprint of life is flexible. And if it's flexible, that means we can manipulate it. Which brings us to a synthetic tool called PNA, peptide nucleic acid.
This is where mapping becomes intervention. PNA mimics regular DNA, but instead of the normal fragile sugar phosphate backbone, PNA is built with a super tough peptide-like backbone.
Like protein armor.
Exactly. Because of that tough backbone, our body's natural enzymes don't recognize it, so they can't degrade it. PNA is practically indestructible inside the cell, and it is specifically designed to exploit Hoogsteen pairing.
So scientists can inject this synthetic PNA into a cell. It hunts down a specific DNA sequence, binds directly into the groove of the existing double helix using those hidden Hoogsteen bonds, and forms a rock-solid triple helix.
It acts as an absolute physical roadblock. When the cellular machinery travels down the genome trying to read a disease-causing gene, it crashes into this indestructible PNA triple helix and stops dead in its tracks.
Meaning we can use it to literally jam the gears and turn off a mutated gene? We aren't just mapping the genome anymore, we are actively hacking its structure to fight disease.
That is the ultimate promise of all this cartography. The physical maps, the STS anchors, the massive ophigels, they give us the precise coordinates. But the discovery of Hoogsteen pairing and tools like PNA give us the mechanism to actually act on those coordinates.
Which brings this whole deep dive right back to you, listening to this. Knowing whether your DNA features specific single-letter variations or unique restriction cutting sites isn't just abstract biological trivia. It is the absolute foundation of personalized medicine.
Absolutely.
The maps we discussed today, from the painstakingly built Hunting scaffolds to the light-activated diagnostic chips, are the exact tools that scientists and doctors are using right now to tailor your healthcare. They aren't just mapping a generic human, they are building the tools to map you.
And this raises an incredibly important question, something really provocative to consider as we close. If Hoogsteen pairing proves that DNA isn't a rigid, static ladder, but a dynamic, flexible structure that changes shape in response to cellular stress, damage, or regulation.
Right.
And if we can target those shape-shifting moments with synthetic PNA, could the future of genome mapping move beyond just charting the static locations of genes?
What do you mean?
Well, we've mapped the street view and the satellite view. But could the next frontier be 4D mapping? Imagine mapping the real-time 3D shape-shifting states of our genome as we actually experience life, stress, and healing. Not just where the genes are, but what shape they are taking right this second.
Wow. A map that changes in real time as we live. That is an incredible thought to end on. From static explorers sketching coastlines to a live, breathing, shape-shifting atlas of our own biology. Thank you for joining us on this journey. Keep questioning the blueprint of life, and we'll see you next time.